Well, first of all, thank you very much for the invitation. My talk, the charge I've been given is to try and discuss the relationship between the activation of the MAP kinase pathway that Dr. Gulek has already introduced some concepts about, and how that connects to the differentiation state of the thyroid cells. So I thought it would be helpful to give a historical perspective first. The discovery of cellular oncogenes really happened in the 1970s, first of all with the fundamental insights that viral oncogenes that were known to be transmissible were ultimately found to result from the capture of DNA fragments, mammalian DNA fragments by these viruses, and that would then introduce them into cells and result in their transformation. And this ultimately resulted in the discovery of key oncogenes, the first being SARC, and relatively soon afterwards with the discovery of the RAS oncogene in a bladder cancer cell line. With the advent of PCR, it became feasible for many labs to begin screening for candidate genes. And among them, a group in Gustave Roussy in France, as well as by Nick Lemoine in the UK, both found that RAS mutations were actually seen in thyroid cancer. Our group was also, involved in these discoveries. Relatively soon afterwards, the group from the University of Naples discovered the RET fusions in thyroid cancer. They were initially observed by a Japanese investigator, but not fully characterized until a paper in Cell, which described both partners in the recombination event. So the group in Naples was very instrumental then in beginning to try and understand how these various oncoproteins change the expression of thyroid-specific genes. And at that point, it was primarily thyroglobulin, which was being investigated, and thyroid peroxidase. We still didn't know which protein transported iodine at that point. And they found that when they transduced viral oncogenes such as MTOSS, which activates the MAP kinase pathway, they could suppress the expression of thyroglobulin. And they became interested in which were the underlying mechanisms for this, and very important advances were made over that period of time. We jump now to 1998, and this was the first demonstration of the impact of expressing a mutant thyroid oncoprotein, in this case the RET fusion, RET-PTC, and the demonstration that this caused hypothyroidism in vivo. This was done by the group of Dr. Rothstein and then by Sissy Jiang at Ohio State University. And so this was clearly an important advance. Now, jumping to 2003, the year before, a group from the UK discovered the BRAF oncogene in melanomas, and many investigators around the world quickly started to figure out, well, to what extent is this particular mutant oncogene involved in their cancer of interest? We were one of those, and our group, in collaboration with Yuri Nikiforov, who was at that point screening papillary thyroid cancers for other genetic lesions, we reported the high frequency of BRAF mutations in papillary thyroid cancer. And several groups almost simultaneously found the same thing. But the insight that we had at that time was that the mutations of BRAF did not overlap with those of RET fusions or with RAS. And the pathway that they all had in common was the MAP kinase pathway. So we even titled our manuscript that this presented genetic evidence of the activation of the MAP kinase pathway in thyroid cancer. And in a way, our mind was prepared for this because in parallel, we were working in a thyroid differentiated cell line where we had introduced the RET oncoprotein with various tyrosine substitutions in the kinase domain. And these tyrosine residues in the kinase are the ones that couple to the downstream signaling pathways. And the question we were asking is, which particular tyrosine residue was involved in signaling to suppress thyroid differentiation? And we found that the tyrosine 1062, which actually couples to the activation of RAS and MAP kinase. If you deleted that, you totally abolish the ability of the RET fusion to downregulate thyroid differentiated function. And moreover, if we just expressed the native RET oncogene, but we then treated them with MEK inhibitors, we could prevent this. So this then gave us a lot of confidence to connect the relationship between the MAP kinase and the differentiation state. A few years later, this was translated into the human setting when Cosimo Durante from at that point in Villejuif and in Rome, he found that in patient samples that the expression of thyroid-specific genes in BRAF mutant tumors was markedly lower than that in BRAF wild-type papillary thyroid cancers. We then actually did an experiment that for us was actually quite instructive, which was to do this in the setting of activating BRAF in the thyroid glands of mice that rapidly induces papillary thyroid cancer. But when we switched BRAF off, we could find that the iodine incorporation was restored. So it told us that this process could be reversible, and that when we treated these mice with either RAF or MEK inhibitors, we could restore radioiodine uptake. 3 years later, the Cancer Genome Atlas study of papillary thyroid cancer was published. We and many others were involved in the analysis of that data. And as you'll see in a minute, it was particularly instructive and to a certain extent confirmatory, but in much more granularity about this important relationship between MAP kinase and differentiation. And in 2013 was the first in-human redifferentiation study done by Alan Ho as first author from our institution. So you've heard from Dr. Gulek about the Cancer Genome Atlas study and the various lesions that are found. And I realize that in the audience, there are many people who don't necessarily follow molecular biology in any detail. So just to give you a background, what you're looking at here are an oncoprint of 496 patients with papillary thyroid cancer, with each column representing a different patient and each row representing a particular genetic change. And as you can see in this study, roughly 60% of papillary thyroid cancers have harbored the BRAF V600E mutation. The second in frequency were the RAS mutations, with NRAS being by far the more common. And then in addition to that, there were a number of tumors that harbored receptor tyrosine kinase fusions, primarily of RET, but also of NTRK1 and NTRK3, ALK, and others. You could see here that when you look at copy number variations, roughly about 70% of these tumors were perfectly diploid, meaning they had no amplification or deletion events. And I'll be talking a little later about some interesting copy number abnormalities that happen in a subset of these tumors. You saw this slide in Dr. Gulek's presentation, but I will recapitulate a little bit, how this came to be. So the first observation that was made was that when you compared BRAF tumors and RAS tumors, they were transcriptomically different. And so the idea then was to say, oh, well, if you didn't develop a 71 gene score that can distinguish the two, and that's called the BRAF-RAS score, where do all the tumors with other mutations fit, right? So here you have the BRAF-like tumors, and of course, those are mostly BRAF mutant. These are the RAS-like tumors. Those are mostly RAS mutant. But here you have a number of fusions. And you can see that the RET rearrangements tend to be BRAF-like. So they tend to have a relatively high MAP kinase output, whereas those with NTRK mutations tend to be more neutral. On the other hand, other rearrangements, such as PAX8 PPL-gamma, which is shown here, which activates a transcription factor unrelated to the MAP kinase pathway, tends to be more RAS-like, retained differentiation, and as do some of the other genetic events that were found. Here below, you're looking at the transcriptomic changes, and you can see how clearly they cluster differently, with the messenger RNA expression pattern of RAS tumors being quite different from that of BRAF tumors. And if you then look specifically at their differentiation score, and that's what Dr. Gulek referred to as thyroid differentiation score. It's a 16-gene signature representing genes involved in thyroid hormone biosynthesis, which tend to be relatively preserved in RAS mutant tumors, but suppressed in BRAF mutant tumors. So why is this? Well, if you try and integrate the information, when you have a BRAF mutation, BRAF signals in this signaling cascade that was shown earlier, but normally when this pathway is activated physiologically by ligands that bind to receptor tyrosine kinases, they usually activate RAS, and they result in RAF dimerization. So two different molecules of RAF need to couple in order to evoke downstream signaling. In the case of BRAF V600E, it signals as a monomer. It does not need to dimerize. And it's therefore unresponsive to negative feedback, which occurs by preventing dimerization. So once ERK is activated, it feeds back on RAF proteins to prevent their dimerization. And of course, BRAF V600E doesn't care, and therefore the output of the pathway is markedly greater in BRAF mutant tumors than in most fusions and then in RAS tumors where the feedback is preserved. So if you then compare the MAP kinase output illustrated below to the differentiation score, there is a very tight relationship between BRAF RAS score and differentiation state. These are mutually exclusive, if you want to put it that way. So let's explore a little bit about what are the underpinnings of this and what explains this. Well, normally thyroid function is under the control of TSH, as you know. TSH binds to its G protein coupled receptor to activate the enzyme adenyl cyclase. And this signals by this second messenger, which is psychic MP, which essentially is what drives thyroid specific function. Now, when you have activation of the MAP kinase pathway, this actually suppresses the expression of lineage transcription factors such as PAX8, FOXY1, and in their absence, the expression of thyroid differentiation genes cannot take place. But there's also a parallel mechanism in which the activation of MAP kinase actually represses the expression of the receptor for TSH. It also interferes with the ability of adenyl cyclase to generate cyclic AMP. So if you actually treat thyroid cells with forskolin, which stimulates adenyl cyclase, in the context of BRAF activation, they are inefficient at making cyclic MP. And also cyclic MP is less efficient at activating protein kinase A. So when MAP kinase is activated, it is essentially interrupting TSH receptor signaling at various levels. So now you can begin to see why it's so critically important to understand how intense or how strong the activation of the MAP kinase pathway is in terms of how profoundly it'll impact thyroid differentiation state. So let me return then to the Tcga study. And we have discussed already that the tumors in the Tcga have either BRAF mutations or RAS mutations or RTK fusions. And what you're looking at here below is just a focused lens on the thyroid differentiation genes, which are illustrated here. And as you can see, in the context of BRAF, where the MAP kinase activation pathway is strongly activated, the expression of these genes is repressed, shown in green. Whereas in RAS and RTK fusions, the expression is largely preserved, or at least partially preserved. However, there is a subset of BRAF mutant tumors that paradoxically retain thyroid differentiation. So we recently asked what might actually explain this. So Laura Bucay in our group went back to the Tcga study and broke down the patients based on whether they had a low thyroid differentiation score in the context of BRAF or they had a preserved thyroid differentiation score. And what you can see is that these patients differed quite significantly in various characteristics of their pathological state and demographic characteristics. So, for instance, the BRAF tumors that retain differentiation tend to have smaller tumors, less distant nodal metastasis, and a trend toward less distant metastasis. Now, most of these patients, therefore, would be T1 or T1b, you can see the majority of them were that or either T2s, whereas in the context of the BRAF TDS low, they were enriched for patients with T2 and up, and so on. Now, of course, many of these patients, based on current criteria, we would choose not to treat with radioactive iodine anyway. So the fact that they had a higher thyroid differentiation score may not be that clinically relevant. So having said that, we decided to actually do a further analysis. And so this is essentially what we did. Here you have a relationship between thyroid differentiation score and BRAF-RAS score. You can see that there is, when you look at all papillary thyroid cancers from TCGA, there's a very good correlation between these two scores. And in the context of BRAF, that correlation is also retained, but with less significance. So if you break them down, and this is what we did, we looked at BRAF mutant cancers that had a preserved TDS, meaning a value greater than 0, versus those that had a suppressed TDS, a value less than 0. And then we reanalyzed the gene expression data. And what we found is that those tumors that had preserved thyroid differentiation, despite having BRAF V600E, were enriched for a certain number of genes. The expression was slightly higher. As just a validation, you can see that several genes in the thyroid differentiation score are, of course, overexpressed in the TDS high tumors. And one that caught our attention was this cadherin 16. And the reason it caught our attention is that the group of Pilar Santisteban in Madrid had done this beautiful study where she investigated the role of cadherin 16 in the formation of the thyroid follicle. These are data from her paper. And what you're looking at, the discovery here was that the lineage transcription factor PAX8 was required for the development of follicular structure in vitro, and that cadherin 16 was what mediated this. And these are just confocal images, and what you're looking at here is what happens when you actually knock down cadherin 16. So in the control setting, you can see in the panel that I'm showing here at day 5, in green, what you're looking is at the expression of esrin, which is normally present at the apical surface, and then the expression of acetylated tubulin, which is essentially coordinating the actin bundling. So this would be a normal requirement for the formation of the follicle. If you don't have cadherin 16, you can see that some of the esrin is actually on the basolateral surface, and that laminin is essentially partially lost. And you can rescue the formation of the follicle in PAX8 deficient cells by overexpressing cadherin 16. This is illustrated here. So the fact that the TDS high tumors that are BRAF have overexpression of cadherin 16 suggests that these may be some tumors that retain some semblance of follicular architecture. And we confirmed that by looking at the follicular fraction that was actually measured in the Cancer Genome Atlas. The other aspect of this study is we looked at the microRNA differences between the BRAF tumors that were TDS high versus TDS low. And several of these microRNAs actually interfere with nodes in the TGF-beta signaling pathway. So why is this important? Well, aside from the MAP kinase pathway, TGF-beta itself suppresses the expression of key thyroid differentiation markers, including the sodium iodine symporter. And so this is from also a study from Pilar Santisteban's group, and many other groups have shown this, in that if you actually treat cells with TSH, you get a nice robust induction of the sodium iodine symporter, but this is suppressed in a dose-dependent fashion by treatment with TGF-beta. So summarizing this aspect of my talk, the BRAF mutant papillary thyroid cancers that are categorized based on their thyroid differentiation score show distinct clinical, histological, and molecular characteristics. This is a heterogeneity that is not dictated by concomitant somatic mutations, but instead the TDS defines subgroups characterized by distinct gene and microRNA expression profiles that may impact their follicular architecture and the TGF-beta signaling pathway. So the last part of my talk, I'd like to talk a little bit about which patients respond to radioactive iodine. So currently, the ATA guidelines have this series of recommendations on which patients should receive radioactive iodine. Low-risk papillary thyroid cancers, the recommendation is not to treat. This week in the New England Journal of Medicine, the group from Gustave Roussy with a consortium in France did the first randomized clinical trial of radioiodine versus no radioactive iodine in patients with T2 or T1B N0 disease and found no benefit. So now we actually have randomized data to validate this recommendation. But it does recommend that we should consider treating in patients who have parathyroidal invasion, extensive nodal disease, BRAF mutations, gross extrathyroidal extension, etc. However, many of these characteristics that are listed here are enriched in patients with BRAF mutations. So it's legitimate then to ask, are these recommendations reasonable, since we know that most of these patients presenting with these characteristics will have a tumor that essentially has decreased expression of the genes required to respond to radioactive iodine. So I'll skip this in the interest of time. So the study that we did was then trying to answer the question, okay, let's try and find out how well radioactive iodine responds in patients with structural disease, with metastatic disease, in which we could actually measure response by RECIST criteria. So we screened our electronic medical record, and I won't go through the details of this, but we started with a large number of patients. We excluded those with thyroglobulin antibodies, and we selected those that had thyroglobulin levels at any time in their course that was greater than 200. It was 221 out of these more than 3,000. And then we looked at those that had a nadir after radioactive iodine of less than 20. And we found that 34 met these characteristics. And of these, 11 had a RECIST version 1.1 response. That means a greater than 30% or greater reduction in their tumor volume. What we did is a case control with 2-to-1 distribution of patients who had what we call exceptional response to radioactive iodine versus non-responders, and their characteristics were very well matched, with the exception of gender, in that the exceptional responders were enriched for female gender, whereas non-responders were more balanced. These are examples, patients who had very remarkable structural responses. This is the patient who presented with a struma ovari. This is the patient with metastatic thyroid cancer to the lung. You can see the beautiful reduction in tumor volume. The Kaplan-Meier survival based on structural progression was clearly different, with much better responses in those that responded to radioactive iodine than those that didn't, and a trend towards improved survival. And this was their genotype. So this is an oncoprint of exceptional responders versus non-responders. And you can see, first of all, that in the exceptional responders, none of these 8 patients actually had a BRAF V600E mutation. They all had mutations of either RAS, RET fusions, or class 2 or 3 BRAF mutations, which actually signal by RAF dimers. So they're different than the V600E. And had very few coexisting mutations of other pathways. By contrast, the non-responders were enriched for BRAF V600E, and many of them actually did have class 2 or 3 BRAF or RAS mutations, but then they also had coexisting abnormalities such as mutations of epigenetic genes, including the switch-Smith pathway, which we have documented prevents response to radioactive iodine, splicing defects, and DNA damage response gene abnormalities. The other interesting characteristic is when we looked at the copy number, the exceptional responders tended to be copy number quiet. So blue is loss and red is gain of copy. Whereas the exceptional responders, whereas the non-responders tended to have frequent amplification events, particularly of this region, which is chromosome 1q. This is illustrated here at greater magnification. So these were arm level amplifications, which were remarkably frequent in the non-responders. And it turns out that 1q was already identified at the TCGA as a factor involved with worse outcome in papillary thyroid cancer based on TNM status. And what we found was when we looked at the thyroid differentiation score of BRAF tumors with 1q amplification, they were much lower than those that had BRAF with normal copy number at 1q. The effects of other copy number abnormalities, such as 21q, made no difference either in the BRAF or the RAS context in the TDS score. And this is just an example of one patient and the copy number abnormalities in the primary. There were two primaries in this case, one of which had a 1q gain, and this was the one that actually gave rise to distant metastatic disease. Other aspects of the study of interest were that the histology did not make a difference, that this is a PCA plot that just looks at the integrated gene expression levels of these tumors. The exceptional responders cluster very nicely together, whereas the non-responders cluster all over the place. Gender, although it was differential between the two groups, did not appear to make much of a difference. But what did is the thyroid differentiation score, which was low in the non-responders and higher in the exceptional responders. And the opposite was true for the BRAF score, where there's a remarkably beautiful correlation between the two. So finally, when we actually looked at oncogenic signatures, no huge surprise here, but blue means decreased expression of these various biological processes in the RNA-seq data, and primarily the non-responders tended to be more endoderm-like, so more primitive, more progenitor-like. They had more activation of the cell cycle and a decrease in overall adhesion, meaning a tendency to become more metastatic, whereas the exceptional responders had much greater expression of genes involved in thyroid hormone generation and in what was called response to metal ion, which we believe reflects the activity of peroxides. And similarly, the non-responders had a high activation of the MAP kinase pathway and other signaling pathways along those lines. So in closing, what we found is that metastatic thyroid cancers that show major structural response to radioactive iodine are enriched for mutations that constitutively activate RAF dimers. That's RAS mutations, class 2B RAF mutations, and RTK fusions, and are usually diploid. RAI refractory metastatic thyroid cancers were enriched for BRAF V600E, have frequent copy number variations, mostly 1q amplification, and harbor lesions of genes in the PI3K pathway, splicing regulation, and in chromatin remodeling pathways. And exceptional responders have a high TDS and are transcriptionally RAS-like. Non-RAI responders have a low TDS and are BRAF-like. And 1q amplification is associated with a lower thyroid differentiation score in BRAF mutant tumors. So I think I'll stop there. I'm just at my allotted time. I just want to acknowledge the people who did that work. Laura Bukai deserves a lot of credit for the identification of the exceptional responders and non-responders and making sure they were nicely matched. Jnana Krishnamurthy, Mahesh Saxena in particular, was involved in the analysis of the gene expression data and the genetic lesions. My colleagues Ronald Gossain, the pathologist, and Ben Shu, who help us a lot in identifying the lesions and characterizing them, and the bioinformaticians Venkat Seshan and Nick Sochi. And I'll stop here and thank you for your attention.